All right. Okay, welcome to the Friday CBC seminar. I'm honored to introduce our guest for today. It's Dr. Jessica Faul. Dr. Faul is an associate research scientist at the Survey Research Center at the Institute for Social Research, which is a powerhouse for survey and social science research at the University of Michigan in Ann Arbor. She is a Wolverine lifer. If you know about anything about University of Michigan, that's really important. But she received her BA in Japanese language and literature from U of M, and she studied in Japan as an exchange student. But then she went on to earn her MPH and PhD from the University of Michigan School of Public Health. At the University of Michigan now, she's involved in lots of centers. She is an affiliate of the Biosocial Methods Collaborative, the Population Studies Center at the Michigan Center on the Demography of Aging. And she is also an associate research scientist on the Health and Retirement Study, where she spends much of her time and work on making this very large and complex study. If you know anything about HRS, it's a huge study of aging, and she makes it run as smoothly as possible. And so I think we can all thank her for that, because through her hard work, she really makes the research life of all HRS users much happier. Her own research work broadly examines socioeconomic predictors of health and health disparities across the life course, including the integration of genetic data to better understand how genes and environments interact to influence health. She has also led the development of a workshop at the University of Michigan to train social scientists on the use of genomic data. So if this is an interest of any of our audience members today, I'm sure Jessica would be willing to follow up with you and talk about that.
Plug at the end.
Yes. Okay. Great. Back to the presentation. So,
great.
Jessica,
Thanks so much, Allison. That was a lovely introduction. I'm really excited to be here. Being someone who's affiliated with their own population center, it's always fun to visit other ones, especially one that really has pioneered the integration of social and biological data. So, really happy for the invitation. Like all the ways I was told this would work, don't work. There we go. Got it. Okay. So today I'm kind of going to take a high-level view on some things and drill down a little bit where I think it might be interesting to this group. So I'm going to talk a little bit about collecting DNA in the context of a large social science survey. I'm going to give you a little bit of a primer on genome-wide association studies, because I find that it's the basis of a lot of what we do in genetics, and it freaks people out. They think it's really complicated, but it's not. So today you're going to learn what that is if you don't already know, and then you're going to feel super smart when you leave. But I'm going to also talk about life after GWAS. So really, that's kind of where we are now. We have a lot of genetic data collected, and we've learned some things, and there's a lot we haven't learned. So there's room for us to keep going forward. And so I'll talk a little bit about that and kind of what the post-GWAS era looks like. Then I'm going to return back to collection and talk about some of the issues that we're still facing when we think about collecting biological data, and some of the things that we're doing to work on that, as well as sort of what the future holds in terms of biological data and measurement going forward. So it's a lot, and I have a lot of slides. I talk really fast, so we should be okay. But that's not to say that you shouldn't interrupt me with questions as we go through, and we'll have time for discussion at the end as well. So Allison already told you a bit about who I am, so I don't have to go too much into this. One thing that isn't on there and is kind of newer is that I'm now the co-director of the Biospecimen Lab at the Institute for Social Research. And again, that's sort of a lab that we've created to do some of the pilot work that I'll mention at the end of my talk. It's really working on developing best practices for biological data collection in the field. It's something that we felt was lacking, and so we decided to help fill the gap, in part for ourselves, but hopefully for other people too. So a little bit about the health and retirement study, since that is really where I spend all of my time. It's the largest and most comprehensive nationally representative panel study of people over the age of 50. It was created in 1990, and it really was the first longitudinal study of older people that included both detailed information about their economic history and situation and health information. And that sounds like all really straightforward now, but at the time, those things weren't integrated. People didn't always think about that together. We're supported by the NIA and the Social Security Administration, and that's sort of been ongoing. We're a cooperative agreement. And since 2006, HRS has routinely collected what we call the physical measure and biomarker data. So this is everything from your height and your weight, your blood pressure, lung function, dried blood spots, salivary DNA. We do this at every wave of data collection that we have, not always longitudinal in every 2 years, but every 4 years. But this did not come about overnight, and it wasn't a decision that we made lightly. So getting into the biological data world really takes a lot of thought and consideration. Our rationale at the time, and specific for adding genomic data, was that the integration of genetic data and social science data was going to be a big transformation, both for genetics and the social and behavioral world. And I think that that is true. And we had Ed Health to look at really as a pioneer in this field. We had longitudinal measurement, which was something actually fairly unique to studies that had genetic data. We were interested in gene-environment interactions because we were social scientists. We had nationally representative samples of African Americans and Hispanics, which was fairly unusual in the genetic world in the beginning when everything was based on isolated men. I joke, but it kind of is true. And HRS was designed for comparability with other studies. So that allowed the possibility for replication and pooling, which actually turned out to be crucial in genetics in a way that I don't think could have ever expected. But we had a lot of considerations to think about as well. For us, this meant a huge change in mode. HRS had done a lot of their interviewing using the telephone prior to 2006, so this meant that we probably were going to have to spend a lot more money and visit people in their homes, so more face-to-face interviewing. We knew it would be an increased respondent burden to ask them to do all this biological stuff. We were concerned about increased attrition. You know, would this really be the thing that sent them packing? There was increased cost, obviously. We had to think about preserving the confidentiality of our respondents. We now had labs that were involved, that were getting all sorts of biological information. It wasn't just us. And really we had ethical issues. We had to think about what are we gonna tell people about what we're measuring? How much of that are we obliged to tell them in return? And also we wanted to preserve the integrity of our study. We're a longitudinal observation study. We did not want to become an intervention. So if we tell you information about your blood biomarkers or your genetics, you know, what does that change? Does that change your behavior? And is that something that we need to worry about? So a little bit about collecting DNA. When you're talking about collecting DNA and the type of work that we do, you're really thinking about it in only non-invasive ways, so like no brain tissue or anything. So you can think about it in terms of getting whole blood, saliva, or dry blood spots. And I won't get into it now, but basically there's pros and cons to all the different ways, all those 3 different ways, related to the ease of collection, their storage, the utility, and the cost. For us, we decided to go with salivary DNA. It was easy, and there was a handy dandy kit that had already been made that preserved the human DNA component of your saliva and not the bacterial DNA, because that's in there too. So this is the Origin collection kit. They come in different styles. We use this one right here that looks a little bit more like a little canister because it can be easily put on the table. And older people have a harder time producing saliva than younger people, so it often takes several minutes. And we don't want to sit around and wait for it, so we keep asking them questions and doing other things, and then they can just deposit more sample in as they go along. So that's worked well for us. Kids tend to use the more test tube shaped ones because you can hold it up to your mouth. Origin is actually discontinuing my favorite kit, so we are either going to have to get them to keep producing them for us or move to the other style. But you know, essentially it's doing the same thing. There's a preservative in there that you mix with the saliva, and then that's fairly stable at room temperature, which again, for a field study is a huge thing. So we've got to make sure that that sample is stable and can get mailed to a lab without a lot of special handling. So once you have your DNA collected and extracted, you have a lot of options. So when we first collected our material, we had no idea what we would do with it, none, zero. We collected it and extracted it and sat on it for about 5 or 6 years. We knew this field was going to be moving rapidly. We needed costs to come down considerably before we could invest in it in a significant way. And then finally around 2010, 11, we finally did. But DNA is a kind of really fun and exciting world. You can measure a little bit of it, or you can measure a lot of it. So you can do things like measure candidate genes. You know the gene you're interested in, you just want to measure that. You can do what we ended up doing, which is a genome-wide chip. So you're kind of taking snapshots along the genome, both in places that you think there's interesting variation, and also places just sort of like mile markers, just kind of some spots along the way. You can look at the exome, which is the part of your genome that actually codes for proteins. Or you can do full sequencing, which means I'm going to measure the entire thing. Those things stay constant over time. It does not matter whether I get it from saliva, from blood, from red blood spot. It doesn't matter whether I get it when you're 20 or 40 or 60. For the most part, that's going to stay the same. That has huge advantages for us. We're trying to get this from 25,000 people. You're never going to get it at the same age. You're never going to get it at the same time. So that's really helpful. There are other really cool things you can get from this sample. DNA methylation, you've probably heard about. Something like telomere length, you've probably heard about as well. Those things you can also get from these DNA samples, but they change over time. That both makes them super exciting, especially when you're interested in interaction with the social and environmental context. But it also requires multiple collections. If you want to see how they're changing in a person, based on whether they've experienced stress or aging or whatever it is. So the genotyping that we did kind of looks like this. We put our samples on these 96 well plates. They then get transferred to a special genotyping chip that gets read by a really expensive machine that fortunately someone else bought. HRS, we measure 2.5 million sites along the genome. And through the magic of genetic architecture and evolution, we can actually take those 2.5 million sites and impute to 22 million. So there's a lot of correlation across your genome. If I measure one place, I actually probably know 5 or 10 other places on your genome that have certain allele patterns. So we can get a lot of information even when we've measured only a fraction of it. This is the type of data that's used for genome-wide association studies. So, and all a genome-wide association study is a linear regression or a logistic regression. So it's an examination of a specific variant in your genome and whether that's associated with a trait you care about. So for height, for example, it's a linear regression. You have this allele, someone else may have this allele. Is that difference associated with height? That's it. And I'm just going to repeat that 22 million times for every single place along your genome. That's all it is. There's no magic. But it's also very simplified, as we'll talk about. Because I just said to you that there's a correlation structure in the genetic architecture we have. GWAS don't account for that at all, like not even a little bit. So if I measure this place here at point A and it's correlated with point B, probably both of those sites are going to kind of rise up in their independent linear regression because I haven't taken that correlation into account. GWAS is really straightforward and simple. None of that factors in. So a lot of times we feel like this. Like we have a mountain of data. There's 3 billion alleles in your genome. What do I know about what I have? And we're often looking just, for the corner pieces, for something to help anchor us. But it really does feel like we're under a mountain of data a lot of times. So a lot of the work that has been done has come about through what we call this consortia framework. Nobody does this alone, the GWAS anyway. So each study that is part of a consortia runs their own genome-wide association study, and then we meta-analyze those all together. So we're really pooling a lot of our data together. This is where that harmonization comes into play. So this is where it became very important that we had the same traits or phenotypes measured as everybody else. So that means that the GWAS that we can run is really limited by how much data we can pull together. So we can run pretty easily a GWAS on education, for example, because everybody measures education and pretty much in the same way. It gets a lot harder to do something like neuroticism. Maybe we can agree on how to measure neuroticism, maybe we can't. But some of those behavioral phenotypes or some of those more complex disease phenotypes get harder to do a GWAS on because we have fewer data sets that have the same information collected. So the harmonization turned out to be an important part in why studies like AdHealth and HRS and some of these players that have a lot of measures often get pulled into these types of studies. So these are really the identification studies. The GWAS are what identify which allele is associated with which trait. And then we move on from there. So this is what the results of a GWAS look like. So this is what's called a Manhattan plot. And this Manhattan plot is just looking at the association between an allele and an outcome, and it's mapping the significance. but kind of in reverse. So higher means smaller, but better, stronger. So it's the p-value. So we need things that reach a p-value of 10 to the minus 8 in order to be significant. And so we're looking at for those peaks. So this is supposed to make you think of a Manhattan skyline, so blur your eyes, imagine skyscrapers. But we're looking for when the significance kind of raises at that level, to that high level that I've circled. So this is cognition. So we knew that we were going to find APOE. So APOE is one of those alleles that we already know is associated with cognition and Alzheimer's disease. So we expected to see APOE and its partner Tom40 down there. And then, but we also found some other sites. We also get this piece of information, which is a QQ plot. And this shows the p-values from all 2.5 million SNPs that we ran. If the SNPs had no association with our trait, you would see that black line follow the predicted red line. And that would just be that it was null. There was no association. But we see the black line, which is every SNP mapped, and pulling away from that null hypothesis. So we can see that, yes, there are several SNPs that are associated. But what this also tells you is that, you know, this is really what we say is highly polygenic, meaning it's not one genetic region that's determining a trait. It's actually a lot of them. And you can see that even though there's like those 3 regions that I circled, there's a lot of this other stuff going on too. And that's maybe not significant at the genome-wide level, but it's definitely worth looking at. I mean, it's different from something that's, say, down here at 0. So, you know, this tells us that most of these complex phenotypes that we're interested in have what we call is a highly polygenic model of inheritance. It's not one thing for the most part that's determining an outcome. It's actually several things that are, you know, have a lot of interplay together. So we've done a lot of these, like a ton of these. I was just telling Allison, it was the part we didn't know we were going to have to get into. So someone wants to organize a consortia that's a, you know, about cardiovascular disease, and then we all kind of have to do our own data analysis and pull it together and give it to them. But that's a lot of work when you have a lot of traits that you've measured. So it's something that we did not necessarily expect and spent a lot of time doing. So we were happy to contribute, but it was a lot of work. So you'll see there's a lot of papers, and they will list all the cohorts that are part of it. So HRS has actually been a part of quite a few of them, which has been exciting. But this stuff changes over time too. So I should probably update this slide so just so you can see how big these are. The GWAS in general tend to be very, very, very big. So when we first measured this, we thought that our sample alone was going to be big enough for a GWAS. We thought we're going to have 12,000 people in our very first genetic data release. People could just subdivide. They could do 6,000 in their discovery sample and 6,000 in the replication sample just using HRS alone. We were very naive. We didn't, I mean, we just didn't really think about it all the way through. You know, if you think about it, you're doing 2.5 million, maybe 22 million independent tests. You need a way bigger sample size than 6,000 to 12,000. But again, we thought the genetic effects were going to be really big. We thought that you wouldn't need that many necessarily, but that's not really what it turned out to be. So way back when we started, you know, there were some big GWAS that maybe had 150,000 people, but over time those got much bigger. And if you wanted to be published in Science or Nature, you were going to have at least 300,000 that you needed. And now those numbers are even bigger. So I think the last educational attainment GWAS had like 1.5 million people in it. You don't necessarily need to be that big, but the bigger your GWAS is, the more powered it is, the more you're going to find significant alleles that are associated with your outcome. And you can see here that, you know, HRS, even though we think we're big and awesome, like we're a fraction of what goes into these studies. And now you may have heard there's a study called UK Biobank. They have released their genetic data, and they alone have 500,000 people who have been genotyped. We'll talk a little bit why, you can imagine why that's awesome, because that's a really big sample size. If they have your trait measured, it's really well powered. We'll talk a little bit later about why that might be a problem. So what has GWAS delivered? So GWAS has definitely identified some genetic variants that are significant and replicated. It's evidence of association with social and health outcomes that we care about and behavioral traits. we pretty much confirm for most things that genetic inheritance is highly polygenic, not just one trait of one allele that's determining or one gene that's determining an outcome. But we've also confirmed that, the 20 years of history that we had of using single genetic variants or candidate genes really doesn't capture the complex and dynamic nature that's happening with us biologically related to our genes. So the promise, we really thought that we would be able to tell everyone who was going to get heart disease and who was going to have cancer and all this kind of stuff from this, and we can't. The genetic effects that are identified in GWAS are still relatively small. I say relative because some things we measure in the social science world are bigger than this and some things are smaller than this. But, and this increases as the size of the GWAS increases. The bigger the GWAS you have, the more power you will have to explain the variance in your sample. So the GWAS we have right now, at best, can explain 11 to 13% of the trait variability that we see. That's educational attainment. But most of the variation we see is not explained for most traits very well. So it's really less than 5% is the R square we get. And here we get to the other big caveat, which is initially, and still mostly to date, most of the large GWAS have been performed in European ancestry samples only. So I joked earlier about the Icelandic men. We're still not too far off from that, unfortunately. And the SNPs, these single nucleotide polymorphisms, which is a word I have not said yet, I've been calling them alleles up until now. It's the same thing. The SNPs that we identify in the European ancestry GWAS often do not replicate to other ancestry groups. So I said before how fabulous it is that the UK Biobank has their 500,000 sample, 500,000 person sample of genotype data. That's great, but their sample is really not tremendously representative if you think about it in the context of our whole world. So they are mostly European ancestry. They are well educated. They are pretty high SES group. And you can think about all the different ways in which that may matter if I'm thinking about, identifying genes that are associated with outcomes in other ancestries that don't have the same genetic architecture, and actually in a different social context. Because my ability to find a genetic association actually depends on the context in which that person lives as well. I'm not going to find the same association in one ancestry group as I will in another, and I'm not going to find the same ancestry group in one social context that I am in another, because there are different forces at play at different levels. So the allele frequency and genetic architecture varies by ancestry, but genes technically have the same biological function. So, does this give me something to go on? So what happens now? So some things we can do with GWAS data. One of the challenges we have is that a genome-wide set of data, 2.5 million, 22 million, pick what number you want, doesn't work well with our social science models. I'm not about to put 22 million variables into my hierarchical linear model. That's not going to work. But unfortunately, a lot of the social science software we have, so it's not compatible with the genetic data. But also, I can't take a lot of the social science ways I think about things. So think about networks or multi-level models or nesting. I can't do a lot of that with the genetic software I have either. So there's a huge incompatibility and a lot of room for improvement there. But for right now, I need to think about simplification or reduction on one side of this equation or the other. So, what I'm here to tell you about is how we reduce the genetic data down. So in general, we can think about using the GWAS data in a downstream analysis in our social science framework in two different ways. One is to aggregate information across the genome. And one example of that is polygenic scores, which I'll talk a little bit about. And another way is actually to think about this gene-based model, to think about, are these significant and replicated alleles or SNPs that I've identified in GWAS kind of marking for me regions of interest biologically that I can look at in a different way with a lot of the social science stuff that I want to. So thinking about gene-based methods, and I'll talk a bit about that too. So my research really focuses, when I'm not collecting data, really focuses on this kind of thing. So looking at sort of genetic and socioeconomic predictors of specifically cognition and memory decline in older adults, and thinking about different ways that we can model these things. So I mentioned one way we think about it as polygenic scores. This, again, is something that sounds really complicated, but is really quite straightforward and easy. We take all these published GWAS that we have, and we basically simplified that down and apply the information we know from the summary statistics from the GWAS to our own data. So we know how strong each allele is associated with our trait, and then we essentially just multiply that out and add it up. It's not sophisticated, it's not complex, it's not taking into account that correlation structure that I mentioned, unless you do that intentionally. But essentially, I end up with what we call a polygenic score, which is a single quantitative measurement of genetic risk. And that's really nice. I can use that in my social science models and be really happy about that. So it's a hypothesis-free measure. It allows us to do things like gene-environment interactions and really maximize our statistical power while taking into account a lot of things that I know across the genome without having to do that individually for every allele. So it's kind of fun and useful, and you've probably seen or heard there's a lot of papers that are looking at using polygenic scores. You should know that not all polygenic scores are the same. They can be calculated in a lot of different ways. In the beginning, we thought we would do something called the top hits, which means just take the alleles that are the most highly associated with your outcome. That for sure must represent all the biological variability we'd ever want to know. It turns out that that's almost never the best method. If best means I've explained the most variance. Better methods, and by better I mean explain more variance, use data across the genome. So where you cut your p-value threshold is up to you. For HRS and other studies, we've all kind of decided to use similar methods so that we are all releasing polygenic scores to our users that are constructed in the same way. And there's a lot more to know about that if you want to. There's also another way of doing it using a program called LDPred. Just to know that there's different ways of doing it. And doing it those different ways means that you will have different association with your outcome, even in the same data set. And you'll have different interactions with environmental variables, even in the same data set. So the way you construct it matters. This is using HRS data just to show that you don't get the same thing in, this is our non-Hispanic white and our non-Hispanic black sample by any stretch of the imagination. So on the right-hand side, we have the non-Hispanic whites. If we look at the SNPs that we have, the alleles that are genotyped, meaning the ones we measure, and we create a score just from those, this shows you what our semi-partial R-squared is. And then this is with BMI. And you can see over here that the numbers I'm talking about are not very impressive. But you also can see that there's a difference between the genotyped and the imputed. So the imputed means I've used all 22 million. So that really is actually adding a lot of noise and not helping us at all. But you can also see that if I use that same set of information and I look at our non-Hispanic blacks, really just compare the gray and the green, the semi-partial R-score is a lot lower in those different ancestry groups. And that's pretty much what we see all the time. It's about a third, no matter what trait we're looking at. Again, because the GWAS were identified in European ancestry samples, they're going to work the best in those samples. So this makes everything we wanted to do, which was, sort of health disparities work, darn near impossible. You can do a lot of work sort of within an ancestry group, but you really can't compare across very well. This is a much more complex way of looking at it. This is a histogram of the density of height polygenic scores. Colors are kind of funky, but in that upper right-hand corner, you can see the distribution of polygenic scores for the European ancestry group in the thing that looks kind of gray-green. And next to it, to the right, is the distribution for our African ancestry samples. So you can see they almost don't even overlap at all. So if you were to think that height, which is actually one of our most genetically determined traits, this would make you think that based on that polygenic score, that European ancestry, people of European ancestry are a lot taller than people of other ancestries. But we know that that's like not at all true. In fact, sometimes it's the opposite. But this tells you that this polygenic score piece of information, is a little skewed and weird and not really giving us a good mapping of, someone's genetic profile for this trait. Down below, I think it's the Hispanic and our Asian samples, and then all together. And you can see that, the reason this European ancestry distribution is sitting way over there is not because they're taller, it's just because we're better at making polygenic scores for that group than we are for anybody else. So thinking about how we move beyond the European-only genetic association, so I just said they're really kind of limited in how much they can inform information about other racial groups. So we've made some attempts to use polygenic scores and gene-level analyses for African ancestry groups in different ways. The way we did it initially was not very predictive or productive. So we really are quite motivated in thinking about ways to do this differently because we are interested in investigating health disparities, and we feel it's important to be inclusive in the research that we do. So one thing that we did, again, in a perfect world, we would have equally large or actually even larger samples of African ancestry and other ancestry groups of people in order to do identification GWAS and kind of do the same sets of things, but in different groups. But we're just not there yet. It just takes time and a lot of investment. So there is a project out there called TopMed, the Transomics for Precision Medicine. Their goal is to actually fully sequence, I think it's more than this now, 120,000 people, including a very large sample of African Americans. This will give us a lot of information that we don't have now. But we're always thinking about what can we leverage in the meantime, because that's going to take a long time. I mean, I think they're almost done, but you can't always get the data very easily. And so one thing we thought about doing is, can we actually take SNPs that are identified in GWAS that are done, in this case, in an African ancestry group, and kind of flip it on its head, use those to identify in other groups, kind of in the reverse way. And some of this is based on those measurement chips I talked about. The measurement chips are actually oriented, the ones that we used, for European ancestry samples. So they were kind of designed to look for variation in European ancestry groups. So if they find variation in other groups, does that inform us about European ancestry samples too? So trying to see if we could kind of back out some of this. So here, we're essentially taking European ancestry GWAS and African ancestry GWAS, building polygenic scores informed by those different sets of studies, and then applying them to our own data. Again, we don't ever, I should say that strongly, we don't ever use our own data to run a GWAS and build a polygenic score from it. It would be, statistically it's bad news. So you always want that identification sample to be different. If HRS has participated in the GWAS, we actually take ourselves out, rerun the meta-analysis, and then use that to inform our summary statistics. So all of these studies, we either removed ourselves or we weren't included. And here we have in blue our non-Hispanic whites, and in orange our non-Hispanic blacks. Down here is where we've used all of our European ancestry-based GWAS. It's the same thing I showed you before. Works really well in our European group, or our Hispanic whites, not so well in our non-Hispanic blacks, across really all the different traits we've looked at. So BMI, blood pressure, cognition, cognitive decline. We were really hoping that this would look very different. So this is where we've taken GWAS of African ancestry samples, created a polygenic score to see how they work. And it's actually worse. And not only is it worse for the European ancestry sample, it's also worse for the African ancestry sample. I mean, that's what they were designed to find that interesting variation in that population, and that still didn't work very well. So if you look for example, here's a BMI score for African ancestry group that I'm only getting, 3.2 in my p-value, and here, here it's better. I have a 5.6. So, that didn't work at all, and we were surprised to see that. So we're not really at a conclusion level for polygenic scores, but essentially what I want you to understand is that the polygenic scores we create from these European ancestry GWAS really don't translate to other groups. We can't use these to investigate racial health disparities. Some of this is, again, because of the orientation of the chips. They're really designed to optimize finding interesting variation in European samples. We need larger non-European samples. So if you ever are on a review panel where someone is proposing to sequence or genotype a group other than European ancestry, please say yes. And we're actually moving toward better chips. So we're moving toward chips that are actually designed for a multi-ethnic sample, which had we had that when we started would have been much better. Or we have to think about doing full genome sequencing for groups like African Americans. And in part, that's just because there's a lot more heterogeneity in non-European populations than there is in European populations, and that's just by way of evolution. So if you're interested in this, there's like a lot of people who are trying to think about methods to adjust for some of that differences in the genetic architecture to still use the GWAS information we have. I'm most excited about what comes from our colleague Patrick Turley, who is trying to create a method of LD score regression that kind of factors in this multi-ancestry differences and maybe can create some scores that we can use across in a multi-ethnic sample. It's called MAMA. It's the Ma and the Ma. It's been in the works for a long time and I haven't seen any results from it yet, so I'm hoping that just means it's more complex than anyone thought, but still is going to work, but who knows. Okay, so I said another way to think about it is to think about the gene. So, you know, we're interested in gene-environment interactions. Let's think about the polygenic score for a while. I've aggregated a bunch of information across the genome. You know, I really shouldn't have any reason to think that the environment is going to work in the same direction across every place in the genome in that way. So a gene-environment interaction using a polygenic score is a real tall order because, you know, here I'm capturing information that's directly related to my trait, but then also other stuff too, like cell differentiation and circulation, and who knows what else is in there that, yes, may be associated with my trait, but I don't expect the environment to be acting in the same way, interacting in the same way. But at a gene level, there's a lot more reason to think that the environment might be interacting with the operation of a gene in the same direction. So two ways we can think about this that's different from polygenic scores and maybe more biologically plausible is thinking about genetic burden scores. And here I'm looking at places along the genome that are in those protein coding regions. They're much less common, because as you can imagine, if I've got a bad mutation in a protein coding area of my body, usually something really bad happens. So the ones that are allowed to exist probably are changing my biology in some way. That's not entirely detrimental to me, because I'm not dead, but you know, they're going to happen fairly rarely. But we think they have a better biological punch. So the genetic burden scores are looking at that. And the sequence kernel association test, or SCAT, is looking at variation in a specific gene region. And we'll talk more about that too. So again, the objective here for this particular project is actually to look at gene-environment interaction from early childhood as well as education on later life cognition, but using these different types of genetic testing. So we do this whole process of how we figure out what genes we're going to look at. So I call this candidate gene 2.0 because in the end, we're still looking at candidate genes. We've just identified them in a way that makes everybody feel more comfortable, which is we let the GWAS determine it. So we look for every single GWAS that has ever been published on a trait we care about. We kind of combine them all together. We have a lot of rules about how big it has to be and whether it's replicated and who gets to stay in it. But in the end, we come out with 39 genes that we think have some biological plausibility to be related to cognition. We looked at the health and retirement study. We modeled cognition in a fancy way with the growth curve model because we were interested in cognition and cognitive decline. We had a lot of variables that we were looking at that were measured in early childhood. So the gene-based analysis, I said, really is just, it's actually just as simple as the polygenic score. It's a count of those rare variants in those protein coding regions, and it's just a score. The more you have, the worse we think it is. So that is what we call a burden score. And when we look at that related to the outcome we care about, so this GB5 score is that burden score, We saw that in our European ancestry group, it was significantly associated with our intercept, so that's our cognitive function, centered at age, I think it was 65 in this, but not associated with our cognitive decline. Moving here is actually whether you had to move in childhood because of financial strain on your family. So we saw that that was associated with our slope. So here, slope is how fast you're declining. So a positive one means you're declining less quickly and negative means more quickly. So that was not necessarily in the direction we thought. And then there was some evidence of an interaction between our burden score and having to move because of financial strain. So that was kind of interesting. We also looked at whether you had to receive help, your family had to receive help in childhood because of financial difficulties. And we saw there was some interesting association there with the intercept, not necessarily the slope. But unfortunately, this kind of model didn't necessarily translate into the same set of relationships for our African ancestry group. Again, we didn't necessarily think we would see it perfectly, but we thought we would see more there, at least for the intercept. Slopes are notoriously difficult to find a lot of really great associations or interactions there. But this was sort of our first foray into trying something different. We made an aggregate measure of financial strain in childhood, and this doesn't look that impressive and dynamic, and it's kind of not compared to other things we do, but for genetics, it's hard to find things. But what we did see is this sort of curve here at the bottom is our group that had the highest genetic burden and a high likelihood of childhood financial strain. So we see that they are our lowest cognitive performing group with the fastest decline. So that was kind of interesting. So one example of a different type of genetic test that you can do. So something else that we did was a sequence of kernel association test. I'm not going to get into this a ton, except for essentially I'm just looking at genetic variation across a very defined region. So here I'm defining my regions as a gene. And we did a lot of, we tested the 39 genes that we had found from the GWAS. And we found 9 had interesting variation, with significant variation associated with this cognitive performance and cognitive decline. APOE we totally expected to find, but some other ones as well. And some of those were in common across the European ancestry group and African ancestry group. So that was reassuring for us. And then we actually did find that education had a marginally significant interaction with several of those genes. So one example of this in the CLPTM1 gene was this, where we found that people who had the minor allele, so the green, here, but the TTs, the greatest risk for low memory performance were people who had less than high school education, who also carried this minor allele. So this helps us, in a way, really kind of drill down and figure out, not just genetic variation across the genome, but very specifically, maybe which genes are at play. We also looked at childhood SES, but in the interest of time, I could get into it too much. But we did find some interesting association there with some of the childhood variables that weren't attenuated after we adjusted for the respondent's own education. So really all of this was to suggest that there are some things that you can do beyond testing every single SNP or allele in a genome or these polygenic scores that may have some interpretability and possibility in sort of a trans-ethnic analysis. And then the things that we're working on right now actually kind of expand this to think about biological pathways. So we can think about genes that are working together in a biological pathway, again, as another way of looking at things at a more biological level that we're hoping kind of translates across different race and ethnic groups. So some other things that we think about quickly, because I do actually spend like 90% of my time worrying about this kind of stuff and only 10% of my time doing the fun analytic stuff, is actually thinking about collection. We spend a lot of time thinking about predictors of non-response and survey data, and also predictors of quality. How do you get a good quality sample from a population-based survey? This is not something that the analytic labs think about at all. That's not how they're optimized. They're optimized to really figure out, a patient comes to a lab, they get their blood drawn, everything is analyzed right away. That's not at all the world in which we live in when we're running sort of a large-scale field-based study. So our goal is to figure out how we can do this better. And one interesting thing we found related to genetics was that we looked at our genetic samples in terms of the volume of DNA that we got and the concentration, which is really the more important piece of DNA that we had. And we found that 25% in our variance of DNA concentration was actually determined by interviewer fixed effects, and only 6% by individual characteristics of the respondent, which means that if you have a really well-trained interviewer who follows your protocol, you will get a really good sample. If you have an interviewer that's not following your protocol, it will greatly affect the quality in terms of concentration and volume of your DNA sample. You know, so yeah, go ahead.
But that's for saliva, right?
That's for saliva. Okay. This is also, yeah, so this was in saliva that we did this. Yeah, so this was actually looking, it's always something we talk about publishing, but we haven't done it yet. So this was actually looking across several different studies. Don't think Ed Health is in there, but it was Wisconsin longitudinal study, fragile families, and HRS, and the Army Stars project. So different studies, different protocols, but interviewers turn out to be a really strong component of that. Something else we spent some time looking at actually relates to telomere length. I didn't really talk about it at all, but you may have heard it's sort of the protective end caps on the end of your chromosomes that protect it when the cell replicates. And the idea is that, you know, as cells divide, telomeres get shorter. It's a marker of cellular aging. You know, when that assay was conceived of and created, you know, this extension to field studies was really not ever considered. So we have longer delays between the time of collection and DNA extraction, and a lot of these fundamental issues about how stable is that sample were kind of left untested. So we designed a pilot to test, you know, how stable is that saliva sample at room temperature before I extract the DNA. I told you those Origin kits are amazing. They are. They let us get the sample to the lab. For DNA, there's no, I don't worry about it at all. Like that DNA is stable there. I probably could put it in an oven, it might still be stable. But for telomere length, these things that changed, we really hadn't tested how stable is that sample over time. Is the quality of the telomere material changing? So we did a pilot where we collected a bunch of Origin kits from bunch of people at the same point in time. We aged them for different amounts of time between 1 and 12 months. What we saw we didn't really like, so we have to replicate it again. But essentially, you know, here you see the DNA concentration by storage time in months. And at 12 months, the concentration of DNA looks a lot different than it did in my 0, 1, 2, and 6 month samples. And unfortunately, that meant that my telomere length actually looked a lot different too. So the telomere length is pretty dependent on the quality of the DNA I have in my sample. So again, these are, you know, these should look all the same. They're from the same people, collected at the same point in time. But my measured telomere length really dropped a lot at 12 months. And again, this has everything to do with how well my sample was collected and stored. But this was something we didn't necessarily expect. So we're going to replicate this again. We really want to be sure that that is the result. We can make good recommendations to people. And we have a new pilot that we're working on, which is both to replicate that one and actually extend this to blood and look at blood collection. Here we're talking about days, not months. No one lets their blood sit out for months. But we do sometimes need often a couple days, maybe refrigerated to get it from the field to a lab. So we're looking at lots of different things related to telomere length, related to freezing the blood, related to freeze-thaw cycles, and comparing it actually as well to the saliva. And we'll do this for telomere length, and we're also looking at it for methylation. So DNA methylation is thought to be fairly stable in the blood, but different cells in the blood may last for different amounts of time, and that could impact the DNA methylation that I get from a pooled sample. So we're doing lots of work to figure out how stable methylation is too. And then just since I mentioned methylation, just some things that we're kind of thinking about going forward, sort of what is the future of genetics for a study like ours, really has to do with epigenetics and social genomics. So the DNA that we talked about today is interesting, and it's really the code for your cellular behavior, but none of that happens unless the genes are expressed. So here we're really interested in thinking about socially sensitive genes that really are going to have a lot more, there'll be a lot more influence there from our social environment. And so that requires that you've measured the social environment throughout the life course in ways like PILOT and ANALYTIC. And here, more than before, more than I talked about with UK Biobank, and more than the European ancestry issue with DNA, having representative samples will be hugely important because we need that variation in the environment. We need that variation in the social and behavioral world. So we're looking at things like mitochondrial DNA, particularly thought to be important related to aging. We're looking at DNA methylation. We're looking at sequencing your RNA, which is just another approach to looking at gene expression. All of these things are dynamic, well, not the mitochondrial DNA, but the other ones, dynamic, change over time, going to look really different at different points in the life course, going to look really, really different in different social contexts. We're going to have a lot of work to unpack this, so it's really exciting that we're all kind of getting on board here. And then we're all harmonizing. We're all trying to give you as data users very similar products so that you can do the work that you want to do. But really, just to sort of wrap it up, what I hope you understand here is that collecting biological data is not magical. It's not a silver bullet. Just because it's biology doesn't mean that we've measured it perfectly. And a lot of the things that we know from survey research, things related to measurement error and non-response, all of that applies here too. Oh, and the importance of a representative sample, all of that applies in the biological data world too. And when we got into this, I think a lot of our social scientist friends were concerned about, what does measuring genetics, what will it tell me, and what does that mean about the social environment? And what I can always tell them now is that, the social and behavioral world matters. It matters a lot. Genetics does not tell us everything. And that really is true of a lot of biological indicators. A lot of them can't be interpreted well without understanding the social context. And so there's a lot more work that needs to be done in order to kind of integrate these worlds and understand how we model that important variation that we care about. How do we represent the environment well in the models that we have? So I say it all the time, I'm like a broken record, but population representative samples, perspective, longitudinal designs, really good measurements of the behavioral and environmental context are really critical to how we understand how genetic risk manifests over time. And here I always say, social scientists, we need you. You're really well positioned to make significant contributions in the translation of genetic risk. Because I'll tell you that in the early days, geneticists didn't think about this. They thought someone's DNA is someone's DNA, and it doesn't matter where I got it from, as long as I have a lot of it. That will just kind of smooth out everything. And that hasn't happened. We really need to think about kind of a representative population approach to science. And this is not a science of short-term gain at all. You've seen we've been in this world for the last at least 10 years. We are still working on concepts and methods and programs to work with the data we have, let alone the data we know is coming, that is going to be exceptionally more interesting and more dynamic. So, I talked with Adam earlier today, people who are thinking about kind of new and exciting different ways to model what we have, thinking about innovative methods. There's a lot of room for you if you're interested in that type of thing. If you're interested in that type of thing, if you're a social scientist who kind of wants to get their feet wet here, we do have a course that Allison mentioned that's sponsored by the NIA, but it's not only applied to aging, it's really anybody. It's a one-week workshop we do at Michigan where we give you an introduction to what genomics is and how we think about it. And again, it's a way to familiarize yourself with the type of data that we have and get your hands on. So it's a hands-on, there's a lab component, you get to work with the genetic data, so it's not so scary and intimidating. We just got supplemental funding to actually design a new course on epigenetic data, so that will be coming in 2020. And we have a website, and you can contact me if you're interested. Applications for next summer's course will be posted in November. I also just want to acknowledge that none of this happens in a bubble. It's never me by myself. Lots of collaborators at U of M, other collaborators elsewhere, and a lot of generous funding from mostly the NIH to do this kind of stuff. both in terms of the pilots and in collecting and distributing and analyzing the data that we have. So that is it. I almost made it.
Like the gene-based setup that you had where
Yeah.
you used the GWAS catalog to define what genes you're actually going to look at.
Yep.
Do you have any sort of measures in place so genes can vary in size
Yeah.
by 20-fold?
Yeah.
And so the number of SNPs that could be contained in a large gene could swamp the number of SNPs in the small gene. So do you have any sort of, first of all, normalization for gene size? And second, there's pretty dramatic differences in the distribution of genes. And so you may find SNPs that could hit 10 different genes or SNPs that are out in the middle of the desert.
Yeah.
Do you have any sort of corrections?
So unfortunately in SCAT, there's not really a correction for the size of the gene. So yes, you're way more likely to find variation across the larger ones than you are the smaller ones. It's just sort of the way it goes. In terms of the overlap, I mean, we did the test independently. So to the extent that, there could be regions that have a bit of a crossover, that crossover just gets analyzed twice. It's not that sophisticated. I mean, that's what I'm saying is that there's really, and that's the SCAT model. The iSCAT is what allows us to look at an environmental interaction with that variance. And that took like years to come out in between, like when we started. So, that's part of the trick is that, in order to take into account all the really complex things about genes, like in terms of size and crossovers, and let alone interactions with other ones, like gene-to-gene interactions, like we're not even close to figuring how to model that. So that's what I'm saying. There's a lot of room for someone probably smarter than me and with a different set of training to figure out this really sophisticated modeling that's going to happen in order for us to really capture a lot of that dynamic interplay and not count things twice and deal with the correlational structure. So no, those things that you mentioned don't get accounted for in SCAT. Maybe in the future. Yep.
So thank you. This is a really interesting presentation. I'm really curious about the ancestry definition. So like on one hand, if you go far back and we're all African.
Yep.
On the other hand, given like racism and the social construction of race and these long histories of colonialism across basically everywhere, not Europe, like when you're thinking about collecting more diverse samples and you're thinking about like one drop rule, in the US, how do we do this? How do we define this group? Okay, so this is an interesting and changing thing too. So I don't have a picture of it, but essentially there's something, we use principal component analysis to help us. So the principal components in genes tend to first discriminate based on ancestry. So the first principal component tells you pretty much how African you are. So how closely associated you are with, what we say is a reference sample from Yorubans in Nigeria. And people will have their principal components cluster around there. And European ancestry people tend to cluster in a different place. And we look at many principal components, but they essentially form clusters. It's a scatter plot, right? So there are clusters, but then there are people who don't tightly fit in the clusters. In order to create an analytic group, we take everybody who self-identify, so you already can see where this starts to go crazy. People who self-identify as a particular race and the cluster, and kind of draw a circle, and how big we draw that circle kind of depends on your own sample, and how big you're going to allow that circle to get. So European ancestry groups tend to be a pretty tight circle. African ancestry groups tend to be a little more bigger of a circle. But there is a ton of people, and in our sample, there's thousands of people that we don't put in these analytic groups that are, just beyond that arbitrary circle that we drew. And these are multi-ethnic, truly multi-ethnic samples. These are people who, have mixed ancestry. And they deserve to be studied too. It's just way more complicated, and we haven't quite figured out exactly how to do it, but we are moving in that direction. So So Hispanics actually gave us our first big challenge. They tend to be a very, very heterogeneous group of people, culturally, your own self-identification, culturally, behaviorally, but also ancestrally and genetically. So we had to kind of figure out a way to do that without really drawing a lot of circles, independent circles, however you think about them as a very heterogeneous cluster. And I think that that's kind of how we're moving to models that allow us to do more multi-ethnic GWAS, to be inclusive of people who don't fit in our tightly defined analytic groups. But it's a moving target, and no one really knows the right way to do it. And in a sample that comes from the United States, for example, we have way, way, way more multi-ethnic and multi-ancestry people than other countries. Not everyone, obviously, but we're going to have to figure it out. But yes, it's a good question about how we make those definitions. And the way we make those definitions could be different than the way somebody else makes those definitions. So when we're talking about replicating, or that, I found this in my sample and we didn't find it in theirs, it could entirely be because they defined their analytic ancestry groups differently. So it's a good question and something that's constantly in flux that we revisit often. Yeah.
Thanks so much. My question is, are there GWAS data that are explicitly based on individuals who have been followed over time?
Yes.
Okay.
Yeah.
So just noting, you mentioned that you can't really study HRS in your own samples,
Yeah.
which are longitudinal.
So there are a couple, but here it's just a sample size issue. So you have to have the data to support the GWAS. So there's definitely been a GWAS of cognitive decline, but it's really tiny. And there's some other ones of phenotypes over time, but it's just about having a large enough set of cohorts to contribute to it. UK Biobank will probably start contributing to some as they have more longitudinal data. So yes, they exist, they just tend to be small and very limited.
Have there been any comparisons of how well explicitly following someone over time compares to simulating it based on?
Yeah, so in one of those examples, we actually used the cognitive decline GWAS to create a polygenic score to model cognitive decline. It didn't work very well. But in part, that's because I know that study had to make a lot of generous allowances for how they measured cognition. Because in order to get people who had longitudinal measures meant that they couldn't be very specific about what cognitive tests went into it. They just said, you have to have 3 domains. You pick which 3 domains you have and give us those scores longitudinally. So I could have very different cognitive tests as another cohort that contributed. So already you can see I've got a really heterogeneous phenotype. It's probably not well defined, and so I'm not going to find the greatest, most interesting genetic association that may or may not translate to another cohort. So yes, they exist. They're really hard. You can't have tight phenotype definitions on them if you do, but over time we might get there. And like I said, UK Biobank will change all of it if you care about European ancestry, rich, educated. That's my bias. Anything else?
I think we take one more question.
Okay, that was great, Jessica. I really wanted to applaud you for focusing in particular on the representativeness of the samples, because as we sit around at these genetic conferences, that's something, and just to emphasize the point about what you're saying about the UK Biobank, that it's all white and high SES, but for this audience, the response rate was 5%. So this is a volatile, so people were asked to contribute and only 5% did. So that also tells you something about representative.
And don't be wooed into anything different when you see the publication that just happened this week about how representative it is. So they did some studies that looked at like mortality risk and cardiovascular risk, and they compared it to the more representative UK studies. That could all and well be true, that you could have kind of similar mortality and cardiovascular outcomes, very narrowly defined on the 3 measures that they represented. But I still am not buying it that means that it's generalizable for everything else we'd ever want to do. So yes, come back to the 5% sample. I mean, again, it's not to say it's not informative.
Right.
You've just been trained to think about things differently, and just come back to it and say, the analysis I just ran represents the data I have, not necessarily the truth. Just keep asking questions about where do these data come from and what does that tell me about how generalizable what I just found is.
Yeah, it's definitely still
Yeah,
valuable.
it's still valuable.
Absolutely.
We still have to do it to get somewhere. It's just, you know, it gets us a step or two forward, and we're gonna need some other piece of information to get us a step or two beyond that.
Thank you.
Thank you.